Hello, students. A very good afternoon. So today we'll have a problem-solving session on chapter that is plant growth and development. So before we head on to this topic, so I will do a small technical check. Give me a second. Yeah, I'll do a small technical check. Please let me know if you can see me clearly and hear me well. Yeah. So I have launched a technical poll, and the YouTube students can put your messages in the chat box. Okay. If there is any technical issues, please let me know. Okay. So by taking one question, so in this session we'll be solving up to 15 questions from your lesson, that is plant growth and development, and let's see which are very primary and very important for your competitive examinations. Okay. So here we have our first question. Typical growth curve in plant is, so we have four options here. Okay, so I'll give you 1 minute to answer this question. So I'll launch an MCQ poll. So you can use this to answer your questions. Solve this question. Okay, students. So we have 30 more seconds. Fine. I'll end the poll now, assuming that you have your answers. Okay. So let us see. So they're asking, what is the typical growth curve? So we know that what is growth curve. You know, any cells or tissues or any whether it is in plants or animals. Okay, with respect to time, right? So when we talk about growth curve, so whenever we see how the growth occurs in plants, so if we see the plant growth, initially what happens is, if you plot it against the time, right? If you plot it against the time, initially what happens, the growth is very, very less, okay, which we call it as a lag phase. And then after the completion of a lag phase, the plant growth will be very, very high, correct? So this will go to exponential phase, and then they go up to stationary phase. A typical graph indicates an S-shaped graph that is called as a sigmoid curve. So with the sigmoid curve, what we can understand is the plant shows three typical phases in its growth cycle. In the first phase, okay, which we call it as a lag phase, the cell division is quite, you know, very slow, and the number of amount of cells are formed. Now the plant enters into an exponential phase, or a log high amount of growth seen. So as it divides and divides, some of the plants cease into maturity, and some of the plants, because of the low supply of the low availability of the nutrients, or the low supplementation of the nutrients, they cease to grow or divide or develop. So this question is D. Okay. So let's see the second question. Opening of floral buds into flowers is a type of. Okay. Autonomic movement of variation, paratonic movement of locomotion. Okay, so option, you can answer your question through polls or through your chat box from YouTube. Okay. Read the question carefully and you have 1 minute to answer this question. Okay. So I will end the poll now. So let us see what our question is. So opening of the floral buds into flowers is a type of what they're asking. Okay. So we have very important growth factor, very important moments of growth when it comes to the flower buds opening into flowers. So if you observe this, we know like there are two different types of moments. One is hypotonic and other one is epitonic type of growth moments. So if you see the epitonic growth movement, now what happens here? The leaves or the sepals that are below, right? They grow faster compared to that of the apical parts, right? So here it leads to the elongation of the leaves, okay, towards the upward direction. Now, when many examples for autonomic movement of growth, okay, which includes epitonic movement of growth, which helps in elongation of the sepals and opening of the petals. Okay, so the option is C. Next one. Next question is, if a tree flowers thrice in a year, that is in October, January, and July in northern India, it is said to be photosensitive but thermo-insensitive, thermo-insensitive but photo-insensitive, photo and thermosensitive, and photo and thermo-insensitive. So students, I'll launch a poll. You can answer this question. All right. So you can answer this question. If a tree flowers thrice in a year, October, January, and July, so those are said to be. Okay. Okay, students, I'll end the poll now. Your time is up. So let's see the question now. See, they're talking about a tree that flowers thrice in a year. That is, one is in October, January, and July. So that too, especially they have specifically mentioned it is northern India, right? So in these 3 months, we face extreme cold, isn't it? The people who live in North India must have a very good experience. These 3 months are winter season, and the amount of sunlight that is received is very, very less, as well as the temperature is also very, very cold. So now, whenever we talk about the sensitivity, if I tell it is photosensitive, that means it is sensitive to light. If I tell it is thermosensitive, that means it is sensitive to temperature. Insensitiveness is it does not react to both photo or light or the temperature. Now, if a tree flowers thrice, especially in a winter season, that is October to July, where there is less light and less temperature, then there is what insensitivity towards both the cases. That means our tree, what we're talking about, is both photo and thermo insensitive, isn't it? So that is why our option is D. Okay, our option is D. Where is the stencil? Okay. Oh, sorry. Yeah, our option is D. Okay. Anyways, we have our next question. Phytohormones are chemicals regulating flowering, chemicals regulating secondary growth, hormones regulating growth from seed to adulthood, regulators synthesized by plants and influencing physiological process. Okay. So people, you have your question, right? So I will relaunch a poll. So you can answer it within a minute, or you can use chat box too. Okay. Phytohormones are chemicals regulating flowering, chemicals regulating secondary growth, chemicals regulating growth from seed to adulthood, regulators synthesized by plants and which influences physiological process. So here is the question and options. Okay, students. So I will end the poll now since the time is up. Okay. So let's see what is our answer. So phytohormones are what? Basically, they are kind of secondary metabolites which are synthesized by plants in a very small quantities. So these phytohormones, they play a very major role in so many aspects of plant growth and development. For example, reproduction, longevity, aging, okay? And all these processes are controlled by these phytohormones. So they are basically the plant growth regulators which are synthesized by plants, which influence many of the physiological processes. Okay. So our option is D. Okay. So what are phytohormones? Phytohormones are a class of secondary metabolites which are synthesized by plants. Okay. And they are also growth regulators. They influence many physiological processes like reproduction, okay, flowering, longevity, okay, and aging of the tree, etc. Okay. So we have our next question. Which of the following is not an inhibitory substance governing seed dormancy? Okay. So I will launch the poll. Yeah, I will launch the poll. Please answer this question. So we have our options for this question, like which of the following is not an inhibitory substance governing seed dormancy? Our options are abscisic acid, phenolic acid, okay, para ascorbic acid, gibberellinic acid. Okay. Okay. I will end the poll and let us answer this question. So the question asked is which is not an inhibitory, okay, which does not inhibit, okay, seed dormancy. That is the question. So if you see the options here, the abscisic acid and phenolic acid and para ascorbic acid, they are all they all govern in inhibiting seed dormancy. Whereas when you talk about gibberellinic acid, okay, gibberellinic acid do not inhibit. Okay, it's not an inhibitory substance. All of this, okay, all of this, they participate in governing the seed dormancy. Understood? So they help in inhibiting seed dormancy, whereas when you talk about gibberellinic acid, it do not participate in that. Okay. So our option is D. It's a very direct question. Next one. Name the plant growth regulators which upon spraying on sugarcane crop increases the length of the stem, thus resulting the yield of sugarcane crop. So this is your question. And then we have options A as gibberellin and B as ethylene, ascorbic acid and cytokinin. Okay. So I will launch the poll again. So you have your poll and you have 1 minute to answer this question. Name the plant growth regulator upon spraying on sugarcane the length increases and also the yield also increases. Okay, students, your time is up. Let's see what is the answer. So they're asking which would promote the length of the stem of sugarcane as well as the yield when we spray it. So when you talk about the options like ethylene, abscisic acid, and cytokinin, so we know the functions of this basically. When you talk about ethylene, so which is a gas, it helps in ripening of the fruits and maintaining the color and texture of the fruit. Abscisic acid is also responsible for elongation of the stem and axial growth of the stem and leaves. So when you talk about cytokinins, the elongation of the root structures and the apical dominance, it's all promoted by cytokinin. When you talk about gibberellin is very responsible for basically when gibberellin is used when especially when it is sprayed on sugarcane, they increase the length of the stem because they promote growth of the stem by increasing the longevity of the cells as well as the cell division. So our option for this question is name the plant growth regulator upon spraying sugarcane which increases the length as well as the yield is gibberellin, not the rest three of them. Okay, next one. Removal of shoot tips is very useful technique to boost the production of tea leaves. This is because, so we have our option, gibberellins delay senescence of leaves, gibberellins prevent bloating and are inactivated, auxins prevent leaf drop at early stages, effect of auxins is removed and growth of lateral buds is enhanced. Okay, so I will launch the poll again. You please try to answer this question. Removal of shoot tips is very useful technique to boost the production of tea leaves. This is because. Okay, students, I'll end the poll. Okay, so let us see what happens when we usually, you should have seen the harvesting of tea leaves. They usually take the, the leaves which are very terminal, okay, or at the shoot tips. So now what happens whenever the shoot tip leaves are taken out, the lateral leaves which are below it, what do they do? They produce a lot of auxins. So whenever they produce a lot of auxins, because of the effect of the auxins, now what happens? It will start boosting the production of more and more leaves which are below it. Understood? So that is why whenever the shoot tips are, when the shoot tips are taken out, the leaves which are below the shoot tips, they produce a lot of auxins. So that will enhance the production of more and more leaves to in the coming days. So that is why our option would be effect of auxins is removed and the growth of lateral buds is enhanced. Okay, so that is our option. So next one, in order to increase the yield of sugarcane crop, which of the following plant growth regulator should be sprayed? Okay, so I will launch the poll again. So you have 1 minute to solve the question. So to enhance the yield of sugarcane, which of the plant growth regulators are used or sprayed? Is it cytokinins, ethylene, auxins, or gibberellins? Okay, students, I will end the poll. So let us see what is our answer. So as we discussed, whenever we have to that is used, okay, is gibberellins. Okay, why gibberellin? Gibberellin actually promotes plant growth with respect to the length or girth. Okay, so usually the mature tissues or the tissues which are almost going under maturation, they which are undergoing elongation and maturation. With the action of gibberellins, they start elongating, so that which is influencing the plant growth. Whereas when it comes to cytokinins, cytokinins are responsible for the root and the apical dominance. Methylene is responsible for fruiting. Auxins are responsible for the formation of new leaves, okay, and the new branches on the new stems. Whereas a gibberellin is responsible for maturation and elongation of the parts. So that is why whenever it is spread on the sugarcane, it helps in elongation of the sugarcane crop. Okay. Next one. It takes very long time for the pineapple plants to produce flowers. Which combination of the hormone can be applied to artificially induce flowering in pineapple plants throughout the year to increase the yield? So we have an option. First one is oxin and ethylene. So I will launch the poll. So you can try to answer this question. Okay. So here is your poll. So students from the YouTube, you can use your chat box. Okay. Okay. Can I end the poll now? Yes. I'll end the poll. So let's see what is used to make the pineapple produce flowers at a faster rate. Okay. So usually the most common hormones or the mixture of hormones that are used to, induce flowering in pineapple is the combination of auxins and ethylene. So it's a direct question, and the option is A. Understood? So usually auxins, the presence of auxins influence, the faster production of or expansion of your floral parts, and ethylene helps in ripening. Okay, so the correct option when it comes to the when it comes leaf drop at early stages can be prevented by the application of dash ethylene, auxins, gibberellinic acid, and cytokinins. So I will relaunch poll again. Okay, so you can answer. Okay, students, I will end the poll. Your time is up. Okay. So the answer for this question is auxins. So what do auxins do? Auxins are very good at preventing the aging. Okay. Aging of the leaves and the composition or the, what do you tell? For example, if you have flowers, the coloring of the flowers and how it ripes. Okay. So the ripening of the flower as well as the aging of the leaves can be actually prevented, and by the application of auxins, and auxins are very very influential in that part. Okay, so this is also a very direct question. So the answer for this question is B. So fine, we have the next question. What causes a green plant exposed to light on only one side to bend towards the source of the light as it grows? Your options are: green plants seek light because they are phototrophic. Next one is light stimulates plant cells on the lightened side to grow faster. Auxins accumulates on the shaded side, stimulating greater cell elongation there. Next one, green plants need light to perform photosynthesis. So here are your options, and I'll launch the poll again. Okay, you can answer this question. And students from YouTube, you can just try to type your answers in the chat box. Okay. So can we end the poll and see what's our right answer? So let's go back to our slide. Yeah. So the question is, whenever a green plant is exposed to the light, one side, usually we know that the plant moves toward the light, okay, wherever the light source is available. So what is happening here? Whenever we place a plant in the light source, okay, or near the light source, now what happens? The accumulation of auxins will take place during the near the shaded part, okay, wherever the light is not falling. So at this point, auxins will start accumulating at the shaded region. Whenever the shaded, you know, whenever the auxins accumulate at the shaded part, now what happens? That will lead to cellular elongation at the shaded region. So whenever the cell elongation takes place at the shaded part, that will push the plant towards the light. So it appears, and it not appears, it actually happens where, as the cell elongation is happening at the shaded region, it moves the plants towards the light regions. So that is why whenever we expose the plants to a light towards one particular direction, the plants bend towards that direction, because usually auxins accumulate on the shaded side. So when it accumulates at the shaded side, this will stimulate the elongation of the cells. Understood? So our answer for this is C. You have to remember this. It's very important. Okay. Next question you have is, auxin can be bioassayed by hydroponics, okay, photometer, then lettuce, hypocotyle elongation. Then we have Avena coleoptile curvature. Okay, so we have four options. I will relaunch the poll for you. You can answer this question. Take a minute and answer this question. So we have 30 seconds. Okay. So anyways, it's a very simple question. I will end the poll now. Okay. So let's see what is our answer. So if you have chosen the option D as your answer, you're right. Okay. So the right answer for this question is option D, that is Avena coleoptile curvature. So what is Avena coleoptile curvature? So basically what we will do excision of the coleoptile. That means the coleoptile region will be okay. So if you have a core, we will cut the coleoptile. Now what we will do, containing auxins, and we start placing at one position. So as we place auxins at one position, that will lead to the curvature, or that will lead to the bending of the new parts, or the elongation of the new parts will be towards the opposite direction where we have placed this auxin. Understood? So this will help in understanding the assay or the presence of auxins, right? So this experimentation is called as Avena coleoptile curvature. Understood? So our answer is D. Next one. Through their effect on plant growth regulators, what do the temperature and light control in plants? Apical dominance, flowering, closure of stomata, fruit elongation. Okay, so I will relaunch the poll again. You can answer this question. Okay. Students from YouTube, you can just type your answers in your chat box. Okay. I will end the poll, students. So let's see what is the right answer for us. Okay. So if your answer is flowering, okay. If your answer is flowering, okay, sorry. If your answer is flowering, then you're right. Why? Because one of the major influences of temperature, right, and as well as light is photoperiodism. Okay, so whenever we have photoperiodism, and it influences completely on how much, like whether the plants will produce flowers or not, right? So photoperiodism principally influences flowering. And whenever we talk about apical dominance or closure of stomata or fruit elongation, they can be controlled by other plant growth regulators such as hormones. For example, if you take hormones like abscisic acid, abscisic acid will lead to closure of stomata. Okay, so when we talk about fruit elongation or apical dominance, when we talk, cytokinins will influence them. So fruit elongation, auxins will influence them. But when it comes to flowering, flowering will not occur until there is a sufficient exposure or a very specific exposure of temperature or light. Understood? So that is why one of the major influences of temperature and light on the plant growth is in especially flowering phase. Okay, next one. Flowering depend on cold treatment is cryotherapy, cryogenesis, cryoscopy, and vernalization. Okay, so I will relaunch the poll. You can answer this question. Yeah, flowering depend on cold treatment is. Come on, students, please try to answer. Okay, fine. I will end the poll. Okay. Let's see what is our answer. Okay. So flowering depend on cold treatment is. So the answer for this question is called is D, that is vernalization. So what is vernalization? Vernalization is whenever you expose a plant to a specific temperature, especially cold temperatures, so that will influence some of the development like flowering. Okay, so it's a direct definition for vernalization. So whenever a plant is exposed for cold treatment, it leads to flowering, and it is quite dependent on this cold treatment. Understood? So answer is D for this question. Fine. So we have another question. One set of plant was grown at 12 hours day and 12 hours night period cycles, and it flowers, while in other hand, this on in the other set, what happened in the night phase was interrupted by a flash of lights and it did not produce flower. Under which one of the following categories will you place this plant? Long day, darkness neutral, day neutral, and short day. Okay. So I will try to launch the poll. So let's see if you can answer. Okay. So what happened? One set of the plant was grown in 12-hour day, 12-hour night cycle. And other set of plant, what happened? The night cycle was interrupted by flashlight. Okay. So under which of the following categories will you place this plant? So we have our option as long day, darkness neutral, day neutral, and short day. Okay. Yes. I will stop the poll. Your time is up. So let's see what is our answer. See, whenever the plant was sufficiently exposed for day and night cycles, it flowered. Okay. But when the night cycle was flower, So one of the primary factors that is influencing this plant to flower is the exposure to sufficient amount of no light or darkness, correct? No. So whenever it requires more darkness to influence flowering, that means the place these plants under shorter day cycles. Okay, so that's why we'll call it a short day, correct? No, people. So if I would have exposed more darkness instead of light, probably this plant will flower. So since its night cycle was interrupted, see, when the night was 12 hours and when it was interrupted by flashing of light, then it did not flower. That means it requires short day and it requires longer dark hours. So that is why we place this plant at short day. Understood? So here we come to the end of problem solving video for the lesson plan growth and development. So if you have any doubts and if you have any queries, you can reach out to your faculties, Yudhishthir sir or me or any of your respective teachers, and you can place your doubts. Okay, students. So thank you very much for your today's session. So we'll end the session for now. Okay. So people who are in YouTube, okay, you can place your doubts in the chat box. All right. Fine. Okay, students, thank you. I'll see you in the next class. Okay. Bye-bye. Hello, students. A very good afternoon. So today we have a session on question and answer solving discussion based on the prior examination that you have written. Okay. So here we have our first question. How many given structures are haploid? So ovary, anther, egg cell, pollen grain, male gamete, zygote, and nucellus. So your options are 3, 4, 5, and 6. Okay, I will launch an MCQ poll. You can just answer your question. And the people who are from YouTube, you can do it directly in the chat box. Okay. Okay. So I will end the poll, students. So let us see our question first. So they're asking how many of the structures are haploid. That means it should have n number of chromosomes. So first, when you talk about ovary, the ovary is a very broad structure, we can tell. So we know that the ovary has several cells within it, right? We have antipodal cells, and then we have egg apparatus, and we have central cell with central polar nuclei. So basically the ovary is not haploid, right? So when it comes to anther, anther is again a bigger structure right? So it's when we have, like, if you take a typical anther, it is a bilobed anther. Within the anther we have microsporangia, right? So in that we have microspores. That is our pollen grain. Except for our pollen grains, rest of the structures of our anther is basically diploid. So when it comes to egg cell, so the egg cell is haploid. So I'll write it as N. The pollen grain is haploid, and then the male gamete is, that is again the pollen grain is again haploid. The zygote, which is formed by the fertilization, is diploid, and the nucellus is also diploid, right? So nucellus is a nourishing layer that helps in maturation of your embryo sac, I can tell, correct? No. So now our option is, out of these many given things, we have 1, 2 and 3. Three of the cells are haploid, so our option is A, correct? So let's go for the next question. Our next question is, during respiration, the conversion of pyruvic acid into carbon dioxide and two carbon organic compounds in cytoplasm is catalyzed by an enzyme. Okay. Phosphofructokinase. Okay. Pyruvate dehydrogenase complex, pyruvic acid decarboxylase, and alcohol dehydrogenase, both B and C. Okay. So you have your options. So let me relaunch the poll again. Okay, we have your poll, students. So students from YouTube can answer through your chat box. No problem. So, okay, students, I'm going to end the poll now. Okay. So they're asking which are the, you know, in during respiration, especially in the plant respiration. I mean, like during respiration, what happens? We have a process, the preliminary process that happens is glycolysis. And in glycolysis, what happens? Glucose is converted to pyruvate, right? So what happens here? Glucose, which is a 6 carbon atom molecule, is converted to pyruvate or pyruvic acid, which is 3 carbon atom. During this process, what happens? Carbon atoms are removed, right? So here, whenever there is removal of your, I mean, like whenever the glucose is reduced to pyruvate, during this process, what happens is, okay, first one, the carbon dioxide release takes place. Now here, once the pyruvic acid formation takes place, correct? No. So when we have a pyruvic acid, which is a 3 carbon atom, and when 3 carbon atom has to be reduced again to produce ATP energy by your oxidative phosphorylation, or you have your lactic acid fermentation or alcohol fermentation. So some of the complexes that are involved are, first one is pyruvate dehydrogenase complex. So where the pyruvate is reduced, 3 carbon atom pyruvate is reduced. Okay. And the next you have is pyruvic acid decarboxylase and alcohol dehydrogenase. So these are another sets of hormones, which another sets of enzymes which reduce our pyruvic acid into carbon dioxide, thereby producing ATP molecules. So our option is both B and C. Okay. Next one. Swollen placenta and oblique septum is a main feature of Liliaceae, Solanaceae, Fabaceae, and Cruciferaceae. Okay, you have your options here. I will relaunch the poll. Okay, students, you can answer this question. Swollen placenta and oblique septum is a main feature of. Okay, students, I will end the poll. So whenever it comes to oblique septum, right? So I will give you one of the best examples to look for, that is capsicum. So whenever you cut the capsicum, you can see that the capsicum, within the capsicum, there is a septum that is dividing your capsicum into several, compartments, isn't it? So capsicum is an example for having an oblique septum. And if you observe in detail, you can also find a swollen placenta. Okay. Capsicum that we eat has a swollen placenta as well as oblique septum. So what family does the capsicum belong to? It belongs to Solanaceae. Okay. So usually Solanaceae has this swollen placenta as well as oblique septum. So our option would be Solanaceae. Okay. That is option B. Next one. Morgan carried out several dihybrid cross to study genes that were sex-linked. He performed experiment in Pisum sativum, sweet pea, Drosophila melanogaster, and snapdragon. So I will relaunch the poll. Okay, relaunch the poll. So you can take a minute to answer this question. So you have to remember Morgan's experiment, right? So what is this experimental organism? Okay. I'm going to end the poll. So actually conducted a dihybrid cross using Drosophila melanogaster, isn't it? Why specifically he chose Drosophila melanogaster? First of all, when we take a look at Morgan's experiment, initially what he does is he takes two of the characteristics like eye color and the body color and eye color and wing shape, and he compares it to our Mendelian as well as the Morgan ratios do not match. That is, in Mendelian dihybrid cross, we have our phenotypic ratio as 9:2, 3:2, 3:1, isn't it? But when Morgan conducted a dihybrid cross, it does not come 9:3:3:1, but the ratios keep changing whenever he chose two different characteristics, isn't it? So that is when it later led to the discovery of there is something called as linkage and recombination. So his experimental organism is Drosophila melanogaster. Understood? So our next question. In a dicot stem, phelogen is, okay? First one, stellar cambium, extrastellar cambium, and develops usually from pericycle primary in origin. Okay. So I will, 1 second, sorry. I will relaunch the poll so you can answer this question. In dicot, phelogen is. Okay, students, I'm going to end the poll now. Okay. So what is phelogen basically? Phelogen is also called as cork cambium, right? So when you see the development of cork cambium, okay, which is basically a lateral meristematic tissue or lateral meristem, and this cork cambium is very responsible for secondary growth, I can tell. Understood? So now, if you see the development of this cork cambium, the cork cambium usually develops from extrastellar cambium, okay? Or your phelogen is developing from extrastellar cambium. So our option is B. Okay. Next one. Read the following statements, that is from A to D or 1 to 4 regarding glycolysis. It occurs in all living organisms. Next one. Phosphorylated form of glucose gets phosphorylated again before converting into fructose 6-phosphate. Next one. First, ATP is synthesized during the conversion of phosphoenol pyruvic acid to pyruvic acid. 4 redox equivalents are removed from the 2 moles of PgAl. Okay. So you have your options, that is 1 and 2, 3 and 4, 2, 3 and 4, 1, 3, 1, 3 and 4. Okay. So I'll relaunch the poll for you. Okay. So you study the options carefully and choose which of the following statements are appropriate when it comes to glycolysis. So, okay, students, I will end the poll. So if you observe the options, right? So the two statements, when it comes, it occurs in all living organisms. Yes, glycolysis have to occur in all living organisms. Okay. So glycolysis is a first step where when you see the oxygen dependency, so the oxygen is not involved in glycolysis. So usually there is enzymatic oxidation process will take place in glycolysis. So after pyruvate production only, whether it is aerobic or anaerobic, depends upon its dependency on oxygen. It will either undergo fermentation or oxidative phosphorylation for the production of ATP molecule. So first option is correct. When you look at the second option, phosphorylated form of glucose gets converted into, yes, glucose 6 phosphate, and again it is converted into fructose 6 phosphate. Understood? So second option is right. Then ATP is synthesized during the conversion of phosphoenol pyruvate to pyruvic acid. Actually, it does not happen there. Next one, 4 redox equivalents are removed from the 2 molecules of PgAl. Actually, that option is also wrong. So our answer is A, that is 1 and 2 only. Okay. Next one. Necrosis is due to the deficiency of copper, iron, okay. And then we have potassium, option A. That is next one. Magnesium, iron, sulfur, and copper. Next we have is calcium, manganese, copper, and iron. Okay. I will relaunch the poll. So you can answer this question. You can take a minute. Okay, students, I'll end the poll now. So what is necrosis? Necrosis is usually called as darkening. So whenever you see the leaves that are losing the chlorophyll and it becomes darker and it becomes curly. So this is called as necrosis. So this is a symptom or a deficiency syndrome, you can tell. So whenever there is a lack of minerals like calcium, magnesium, and copper and potassium, usually necrosis occur in plants, which leads to darkening of your leaves. Okay, next one. Find the incorrect combination from the 4 options. Next question. Lycopsida, which involves Selaginella and Lycopodium. And next one, Pteridopsida, that is Dryopteris, okay, Pteris, and Adantum. Next one is Bryopsida, Funaria, and Polytrichum. Next one you have Phaeophysia, that is Ulvox, Fucus, Spirogyra, and then we have Spacum. So out of this, we have to find the incorrect combination, okay? So I will relaunch the poll. Okay, students, I will end the poll now. Okay. So your options, if you observe, okay, when it comes to Lycopsida, Selaginella and Lycopodium are the right examples. And when it comes to Pteridopsida, okay, Dryopteris, Teres, and Adantum, they are also right options. So in Bryopsida, when it comes to mosses, Funaria and Polytrichum are also very correct options. But if you see the last option, that is Phaeophyce, Volvox is a chlorophyll containing, okay, it contains chlorophyll, that it belongs to green algae. So it is not Phaeophyce. So Volvox is a wrong combination when it comes to this. So our option D is incorrect combination compared to all of these four. Okay. So our answer is answer this question. So gametocytes during the life cycle of Plasmodium, they develop in what? Okay, I'm going to end the poll now. So let's see our options. Okay, gametocytes. So what happens whenever we have a malaria? When you take a case of malaria, in malaria, what happens? First, a mosquito bites, right? So when a mosquito bites a human, so basically sporocytes are released into the bloodstream, and that will go to liver, right? In liver, what will happen? They will proliferate asexually, and from there they will migrate to red blood cells. In red blood cells, again they will multiply asexually, and during this phase they will enter a sexual phase where they produce gametocytes. Okay, so usually gametocytes develop in red blood cells of man or human. So after that, these gametocytes will be taken by mosquito again. Okay, so mosquito will take up. So the gut environment of the mosquito will help in the formation of your sporocytes. Okay, and then sporocytes again mature to the salivary glands of mosquito, and then again mosquito bites the human, which new sporocytes, and then the second cycle of infection begins. Okay, next one. In ECG, P wave represents the depolarization of the ventricles, repolarization of the ventricles, depolarization of the atria, and repolarization of the atria. Okay, I'm gonna launch the poll again. Okay. Okay, students, I'll end the poll now. So they're asking about ECG, isn't it? What is ECG? ECG stands for electrocardiogram. So basically, it is performed to understand the normal functioning of our human heart. So whenever we conduct ECG, basically we come across something like we have P wave, and then we have QRS complex, and then we have a T wave. Okay, I'll write it as P, Q, R, S, and T. Okay, so in P wave, what happens is first one, your atria, right? Your atria have to depolarize. So when the atria depolarize, now what happens? The atria will contract, pushing the blood from atria to the ventricles. Okay, so this wave represents the depolarization of atria. Whereas the QRS complex, what happens? QRS complex is very large, very big peak. Why? Because the ventricles depolarize at the QRS complex. Now, if you see what happens here also, so usually during the QRS complex, okay, at the same time, your repolarization of atria will take place during the QRS complex, okay. See, depolarization of ventricles takes place in QRS complex, as well as repolarization also happens during this period, so that we will not get the two waves, we'll get only one wave, okay, it will overlap. Whereas when it comes to T wave is repolarization of ventricles. So our question is about P wave, or P wave happens where depolarization of atria happens. That is when we get the P wave. Okay. Next question. When some structures develop along different direction due to the adaptations to different needs, this is dash evolution, and these structures are dash. Okay. What is the correct options for X and Y? So I'll relaunch the poll. You can answer this question. Okay, students, I'm gonna end the poll now. So let's see your question. So whenever we have same structure, you see this word, same structure and different function or needs. This is called as what? Whenever an organ has same structure, but it performs different function, it is called as what? Homologous organs, right? So homologous organs are an example for what type of evolution? Divergent evolution. What is divergent evolution? If I take example of limbs of human, okay, four limbs of cheetah, okay, and the flippers of whale. Okay, if I take these examples, all of us have the similar four limbs, but look at what we use them for, right? So we use them for different we have evolved different structures out of it to perform different function. Same structure, but different function, right? So the answer for this is dash evolution. It is a divergent evolution. And what type of organs? Homologous organs. So our answer is C. Okay, next one. How many of the given statements are correct? So electrical synapses are rare in our system. Second one, the rise in stimulus induced permeability to sodium is extremely short-lived. Third one, cerebral medulla has tracts covered with myelin sheath. Okay. Then motor end plate is receptor in a knee jerk reflex. The neural organ. Okay, we have the fifth option also. Okay, this is the fifth option. The neural organization is very simple in lower vertebrates. Okay, so I will launch the poll again. So you can take 1 minute to solve this question. Okay, students, I will end the poll now. Okay, how many statements are correct here? So if you have chosen three, right, your answer is right. So which are the three? So first one is electrical signals are okay, rare in our system. So that is actually wrong. So the rise in electrical stimulus, basically stimulus, induced permeability to sodium is extremely short-lived. That is correct. Okay. So next one, cerebral medulla has tracts covered with myelin sheath. Yes. Next one is motor end plate receptor in knee jerk reflex. Okay. One second. Yeah, motor end plate is receptor in knee jerk reflex. That is also correct. Next one, the neural organization is very simple in lower organization. Okay, in lower organisms. So we have 1 and then 2 and 3. Which one? The first one is the rise in stimulus of the induced permeability is very short-lived. That is correct. Next one is your cerebral medulla is covered by myelin sheath. Next one is the neural organization is very short-lived in lower vertebrates. Okay, these are the 3 options. With column 1 and column 2. So you have your column 1 and column 2. That is in column 1 you have embryo of grass, Wilsonaria, castor, and five free segments in syncarpus pistil. And then in column 1 you have dioecious, monoecious, okay, hibiscus, epiblast, and pisum. And you have your options. So I will launch the poll again. So you can take a minute to answer this question. Okay, students, I'm going to end the poll now. Okay. So our option, what is embryo of grass? Embryo of grass is an example for epiblast. Okay. So this is one I can tell. I'll write it as A here. So what is epiblast? Basically, it is a single layer of cells. Okay, I can tell. So in this, what happens is in epiblast, basically, you have a primary ectoderm and an internal layer of cuboidal cells, okay, which are called as hypoblast. So when it comes to your embryo of grass, it is an example for epiblast. Okay, next one, Vallisneria, since it has both male and female, okay, parts, so it is an example for dioecious, right? Next one, coming to castor seeds. Castor seeds an example for monoecious. Okay, I'll write it here. And then we have 5 free segments in syncarpus pistil. Okay, so that is an example for hibiscus. Okay. So when the carpels are fused, that is called as syncarpus, and we can see it in hibiscus. So our option is C. Okay. Next one. So you have here substances that have dash find it difficult to pass through the membrane. Their movement is to be dash. So membrane proteins provide sites at which such molecules cross the membrane. So you have to find what are A, B, C. So I will relaunch the poll for you. You can answer this question. Okay, students, I'm going to end the poll. So let's see. See, our membranes are basically semi-permeable and mostly it is a lipid bilayer. So that is why what happens usually the hydrophilic molecules. If you see the plasma membrane has hydrophobic tails, okay, which are usually in the center or towards the inside. So that is why whenever a hydrophilic molecule has to enter, it does not interact well with this inner membranes. So that is why for the movement of any hydrophilic moiety, okay, they find it very difficult to pass through the membrane, and their movement have to be what? It has to be facilitated. Okay, we have several factors like transport proteins, right? So our transmembrane proteins or channels, protein channels, what do? They facilitate. So these membrane proteins provide site at which such molecules cross the membrane, and they do not set up a. So do they set up a concentration gradient? No, correct. So they do not set up a concentration gradient. So that is why our option is A. Okay. Fine. So we have the main biological control method of crop pest is based on competition, amensalism, predation, and parasitism. Okay. So now I will launch poll again. You can answer this question. Okay, students, I will end the poll now. So let us see what is our answer. So whenever it comes to the control method of crop pest, it's basically what happens is, so the control of pest happens upon prey and predation relationship. So here what happens whenever there is a very proper, okay. So whenever we have a proper balanced prey and predation, what do you tell, ecological interactions. So then what happens, there is a proper way in which the predators drive their energy from the prey, correct? No, they extract their energy from the prey. So this control method, okay, of a predator keeps this population size of the pest under very much control. So if we want to control any pest in, we have to go for prey and predator. So that is called as predation, isn't it? So one of the major mechanisms or methods for pest control is basically coming from predation. Okay, students, thank you very much. We come to the end of this question solving session. I will see you in the next class. Okay, so here we end the session. Thank you. Bye bye.